As part of my research over the past 10 years or so, I look at bacteria that cause infections in humans and try to understand how they cause disease and how we can try and prevent that disease or cure that disease. That type of work typically uses animals at some point in the study. There's huge pressure to reduce the number of animals that are used. I was interested in looking at alternatives to working with mammals. I'd read a couple of papers where Galleria melanella had been used and thought I would just try that. But one of the problems that we had was that the larvae we were using were all fishing bait. We were introducing this huge level of variability into our experiments. So although the results correlate well with what you get in mammals, it's very hard to sort of see differences between things because there's just so much noise in the system. So we set up an inbred, genetically determined, age-defined, weight-defined, surface decontaminated and genome sequenced line of the insects.
We became aware that although this model had enormous potential, there were also some problems with larvae that weren't purpose-bred for research. And that really drove us towards providing a much better quality-controlled larvae that would be available to the community. TruLarv offers a model that they can use to obtain data more quickly, more reliably, and there are significant cost savings as well. It enables them to do this more cheaply. People are finding this product to be really valuable and to really enhance the value of the research they're carrying out.
I've been using bait shop larvae to test many of my strains. I would do one experiment and then follow it up a week later, and I'd find that the results from week to week were variable. When we switched over to TruLarv, I found that this reproducibility was much better. We can do high throughput screening. In my case, I can test hundreds of strains quite quickly and very cheaply. But I wouldn't be able to do that with mammals. I would only maybe be able to do 2 or 3 strains, and I'd have to narrow down my field of research quite extensively.
In terms of easy handling, the Galleria are very easy to use. These larvae are inexpensive to buy. They don't need to be fed or watered. They don't need to have special facilities. You don't need a home office license. They have a nicely defined injection site actually on them so that you can say this is the point at which the infection has been injected. They are quite easy to score as well.
We see that there are 3 main applications. One is in infectious disease research. One is as a model for drug screening, and in particular screening antimicrobial drugs. And finally, what we've seen more recently is to screen chemicals for toxicity. It enables people to do those studies much more cheaply, much more effectively, and to use much larger groups to give much more statistical power to those studies.